Hey guys, today I'm going to talk about glycolysis. So what is it? Where does it occur? How does the glucose reach the cell? And why is it important? So today I'm going to cover all about glycolysis. So let's begin. About the discovery. The process of glycolysis was discovered by three scientists, Gustav Embeden, Otto Meyerhof, Jacob Carol Parnas, from where we got the Embeden, Meyerhof and the Parnas, which is also known as the EMP pathway. So that's why we call glycolysis as also an EMP pathway. So what is glycolysis exactly? Glycolysis has originated from two Greek words. One is the glycise and a lysis, where the glycyle means sweet and the lysis means splitting. That involves the breakdown of sugar, or the process in which one or more glucose is partially oxidized into 2 moles of pyruvate in a series of enzymatic catalyzed reactions. It is a unique pathway that occurs in both aerobic as well as in anaerobic conditions. And the location where it occurs is the cytoplasm. The next question arises is, how does the glucose reach the cell? So as we know that during the process of digestion, which is a complex process, we take in a lot of food, in which the carbohydrates get converted to simpler sugar, like say glucose, and in order to be utilized by the cells, it passes through blood streams. Now, as the glucose molecules passes through the bloodstream, it passes from the extracellular to the intracellular space through a glucose transporter called the GLUT into the cytoplasm, where the process takes place. It is a 10-step process that includes 2 phases. One is preparatory phase and payoff phase. Preparatory phase is also known as the energy investment phase, that requires 2 ATP molecules to generate high energy intermediates, whereas the payoff phase is also known as energy generation, and the intermediates are metabolized, forming 4 ATP and 2 NADH molecules. Start with the phase 1 preparatory phase. The glucose, which is a 6-carbon molecule, is phosphorylated to form glucose 6-phosphate, which is an irreversible reaction and catalyzed by hexokinase. It also requires divalent metal ions such as Mg2+ for the activity. And it also utilizes the ATP in this reaction. Further, isomerization takes place, in which a reversible conformational change of glucose 6-phosphate to fructose 6-phosphate. Further, fructose 1,6-bisphosphate is formed by the addition of phosphate group in fructose 6-phosphate. This irreversible reaction is catalyzed by phosphofructokinase in presence of ATP. Now the fructose 1,6-bisphosphate is cleaved to produce 2 3-carbon molecules. One is dihydroxyacetone phosphate, and the next one is glycerolaldehyde 3-phosphate, catalyzed by aldolase. Now dihydroxyacetone phosphate rapidly and reversely converts to glycerolaldehyde 3-phosphate by enzyme triphosphate isomerase. Now the 2 molecules of glycerolaldehyde 3-phosphate are oxidized further. The enzyme glyceraldehyde 3-phosphate dehydrogenase catalyzes into 1,3-bisphosphoglycerate with the help of NAD. This step is catalyzed by the enzyme phosphoglycerate kinase to form 3-phosphoglycerate. 2-carbon-2 to form 2-phosphoglycerate. Now the removal of water from 2-phosphoglycerate form phosphoenol pyruvate catalyzed by enolase. The last step, the glycolysis. In the last step of glycolysis, pyruvate is formed catalyzed by pyruvate kinase. Last but not least, once the pyruvate is formed, there are 3 metabolic fate of the pyruvates. Under aerobic condition, the pyruvate formed as the final product of the glycolysis undergoes oxidative decarboxylation by a set of reactions which is catalyzed by pyruvate dehydrogenase in the presence of NAD and coenzyme A. Now the majority of the pyruvate is metabolized in the mitochondria under anaerobic conditions such as occur in muscles during vigorous exercise or in erythrocytes which lack mitochondria. Pyruvate is converted to the lactate with the help of an enzyme called the lactate dehydrogenase, in which NADH is being utilized. This process is also known as the lactic acid fermentation. In the third case, in many microorganisms such as yeast, pyruvate is converted to carbon dioxide and ethanol using enzyme pyruvate dehydrogenase and alcohol dehydrogenase. So if you see in this reaction, the pyruvate first gets converted to acetylaldehyde and there is a release of CO2 catalyzed by the enzyme pyruvate dehydrogenase, and further it gets converted to all of the enzymes of glycolysis are found in the cytosol. The breakdown of the 6-carbon glucose into 2 molecules of 3-carbon pyruvate occurs in 10 steps. The first 5 steps of this pathway constitute the preparatory phase, and the other 5 steps constitutes the payoff phase. The 3 glycolytic reactions are irreversible, and these reactions are catalyzed by hexokinase, phosphofructokinase, and pyruvate kinase. As a whole, at the end of the reaction, we get the net reaction in which the glucose is being utilized and it gives rise to 2 molecules of pyruvate. Hope you liked the video. You can also connect us through the other social media platform. Thanks for watching.